Oh, so Christina, who else is left?
That's it. It was Madame.
Oh, nice. Oh, c'est chaud.
Non, mais
Bonjour à tous. Bienvenue à la présentation scientifique d'aujourd'hui. It's a pleasure to welcome Dr. Nikolova. Dr. Nikolova completed her PhD in psychology and neurosciences at Duke University in the US, and then did a fellowship, a postdoctoral fellowship in translational neuroscience at the Center for Addiction and Mental Health in Toronto, where she was then recruited as an assistant professor. Her work uses translational neuroimaging and genetics to study biological mechanisms underlying brain aging and mental illness across the lifespan. Dr. Nikolova, bienvenue, la parole est à vous. The mic is yours, Yuliya.
Thank you so much. Hello, everyone. Unfortunately, I can't see the room, so if anyone wants to raise a question, please feel free to speak up. And yeah, thank you so much for the invitation. It's a pleasure to be here today with you virtually. I gather that this is perhaps the last talk of the year, so I have my Christmas tree in the background for some extra festive cheer. Please accept my apologies for not being there in person. I was unfortunately not able to travel this time around, but I hope I'll have the opportunity to visit your institution in the not too distant future. And in the meantime, I'm happy to share some of our work and hopefully spark some interest in a continued dialogue and some new collaborations, who knows. So the focus of my presentation today is on neuromodulatory systems or mechanisms that lie at the intersection of aging and mental illness. And my lab at CAMH more broadly studies the biological pathways that overlap or converge between mental illness and brain aging, which are these otherwise distinct phenomena. And in recent years, there's been a proliferation of studies, both from my group and of course many others, showing that having a psychiatric diagnosis or mental illness more broadly tends to be associated with an older-looking brain, and in some cases also older-looking peripheral tissues. And this is detectable on different levels. We see it on the level of brain, macro structure. It's also visible on more basic molecular and cellular phenotypic levels, including gene expression and so on. And this is certainly consistent with some epidemiological associations that we observe. For example, again, having a diagnosis of mental illness, some diagnoses more than others, of course, are associated with overall elevated risk for age-related cognitive decline and even dementia in late life. However, as is often the case in our field, the exact sort of biological mechanisms that underlie this link are not very well understood. And what we're interested in doing in my lab is focusing on what we like to call, mechanistically informed biomarkers that can both provide clues as to the underlying biology of these epidemiological links, and also serve as risk biomarkers that could help identify vulnerable individuals so that we can target preventive interventions accordingly. So today, I want to focus on two specific diagnoses or groups of diagnoses, if you will, that have been independently associated with risk for age-related cognitive decline, namely late life depression or LLD and substance use disorders or SUDs diagnosed in adulthood. And again, both of those have been associated with independently with dementia risk and cognitive decline. And what I want to focus on today is two specific, or I guess two respective mechanisms that I believe are promising but understudied mechanisms that might help explain that cognitive risk in those disorders. So specifically, the neuromodulatory systems that I referenced in the title of my talk are the norepinephrine, also known as noradrenaline system of the brain, and the dopaminergic system of the brain, which you can see depicted here, these very sort of classic canonical depiction from every neuroanatomy textbook. So just very briefly, the two systems share some anatomical features. So in both cases, the main effector neurons, noradrenergic and dopaminergic neurons respectively, reside in the brainstem. For the norepinephrine system, that is, of course, in the locus coeruleus, denoted here is the green circle saying LC. I don't know if you can see my cursor, hopefully. And the LC then projects to the pretty much the entire rest of the brain, including the hippocampus, the prefrontal cortex, and really the entire cortex in some sense. The dopaminergic projection system is a bit more focal. The dopaminergic neurons originate in the substantia nigra, specifically substantia nigra pars compacta, which is denoted here on the right as SN, and of course the ventral tegmental area, or VTA. Those also project to subcortical targets and eventually cortex. And again, those systems are fairly well known. There's nothing new here. They've been shown to have key roles in motivation, memory, and executive functioning, which is not surprising given their projection targets. Interestingly, of course, all of these constructs, all these dimensions of cognition, if you will, are also altered across many different psychiatric diagnoses, and they're also very sensitive to age-related change. So what is perhaps a bit less well known is that the LC, the locus coeruleus, and the substantia nigra are also among the most age-sensitive regions of the brain. So that's why today I want to focus on how those regions may potentially mediate the link between late life depression on the one hand with norepinephrine and dementia risk, and then on the other hand between substance use disorders and cognitive decline mediated through the dopaminergic system. So what do I mean when I say that these are very, very age sensitive? There's a lot of hints as to that phenomenon coming primarily from postmortem pathology work. So this is a study that was published back in 2012 looking at autopsies for individuals. Some of them had dementia, others did not. But what was really striking here is that they found tau pathology was present in the substantia nigra and the locus coeruleus of 44% of the autopsies that were otherwise at Braak stage 0, meaning there was essentially no detectable cortical pathology. So to me, this speaks to these nuclei in the brainstem as being really potentially the first sites to start accumulating age-related damage or pathology, if you will. So they may really represent sensitive biomarkers of potential additional risk for age-related cognitive decline. Now, thankfully, we don't have to only rely on postmortem studies to evaluate the health or the integrity of these nuclei. We now have neuroimaging methods. Specifically, I want to talk about the neuromelanin sensitive MRI method, or NMMRI. This has many names in the literature, but for the purposes of today, I'm going to be calling it neuromelanin sensitive. So just very briefly, neuromelanin is a byproduct of dopamine and norepinephrine metabolism. It builds up very exclusively, actually, in the locus coeruleus and the substantia nigra compared to the rest of the brain. It binds iron, which means it has magnetic properties that make it relatively easy to visualize using specialized MRI sequences. So what you can see down here in the bottom left corner of my slide is one such neuromelanin sensitive image showing, kind of hard to see, but they are there. The two bright spots right sort of anterior to the fourth ventricle in the brainstem. So those represent the locus coeruleus. And what you can see though is that there's a lot of just general sort of background noise. So a lot of additional bright spots, not quite as bright as the LC, but somewhat bright nonetheless. So because of this sort of background signal, the main metric that is typically used to index the integrity of the LC, if you will, is the ratio of the brightness of that area to the brightness of a background region known not to have any neuromelanin. And that's referred to as the contrast to noise ratio, or CNR. And I also realize I have a lot of abbreviations in my talk, so I'm going to try to spell those out rather than use the abbreviations whenever possible. So please bear with me. So we have this very handy metric, contrast to noise ratio, which broadly reflects how healthy this the locus coeruleus is, and it's very, very similar to substantia nigra. And there are prior imaging studies that show that this CNR metric, this contrast to noise ratio metric, follows roughly an inverted U-shaped trajectory over the lifespan, where there's an initial buildup of signal, likely reflective of buildup of neuromelanin, happening purely as a result of norepinephrine and dopamine turnover, just by using those structures. And then there's a bit of an inflection point in late midlife, around age 57 in this particular study, where the slope sort of shifts and turns around, and we start to see a bit of a decrease in signal. So this latter part is thought to reflect more neuronal loss. So the initial buildup of neuromelanin is believed to be more or less adaptive, because neuromelanin can also segregate toxic iron species such as iron, but later on, eventually those mechanisms begin to, in a way, sort of get depleted and fail. And once neuromelanin is released from a neuron upon cell death, it can be neurotoxic to surrounding neurons. So essentially, we believe there's like these two different phases of sort of neuromelanin accumulation and then neuronal loss, which can be helpful when sort of tracking the structural properties of these regions. Okay, so just to kind of summarize this somewhat lengthy intro and link this back to late life depression, what we know is that the locus coeruleus, or the LC, is implicated clearly in cognition, aging, and neurodegeneration. And in fact, it may be one of the most sensitive regions to age-related pathology in both normal and more kind of pathological aging. At the same time, late life depression is also associated with elevated risk for cognitive decline and dementia, but of course not everyone who has a diagnosis of late life depression is going to go on and decline cognitively after that or develop dementia. Plus, we don't expect to see huge degenerative changes necessarily in late life depression compared to, for example, diagnosed dementia. So based on this exquisite sensitivity of the LC to aging, and the fact that we're looking for relatively subtle markers of cognitive vulnerability in late life depression, we asked ourselves the question, is the structural integrity of the locus coeruleus altered in late life depression? And a related question, does the integrity of the locus coeruleus associate with cognitive health or cognitive performance in late life depression? So to answer that question, we use data from the SENDEP study, which is led by PI Dr. Breno Diniz, who's currently at the University of Connecticut. This is a NIMH-funded study that recently wrapped up, and SENDEP stands for Linking Molecular Senescence Changes to Depression and Cognitive Impairment in Late Life. And the inclusion criteria for the study are fairly simple: age over 60, currently depressed or never depressed. Exclusion criteria are current dementia. Interestingly, mild cognitive impairment was not exclusionary in this case, but the majority of the participants were cognitively intact. And I should say that the neuromelanin MRI had nothing to do with the overall initial aims of this parent study, but Breno was kind enough to allow me, as an early career investigator back in 2019, to piggyback on this larger study and integrate a neuromelanin MRI sequence as a pilot in a subset of participants. So we were able to actually collect that in 26 participants with late life depression and 26 never depressed controls. And you can see the demographics for those participants right here in the bottom right. The groups were fairly well matched for age, which as I think is pretty important. For biological sex, they were actually not perfectly matched. Unfortunately, we did have slightly more female individuals in the late life depression group, but we ensure that we controlled for that in all analysis, just to make sure that none of the results are driven by confounding effects. Everyone participating in the SENDEP study also completed a series of cognitive assessments, most notably the R-bands, the repeatable battery for the assessment of neuropsychological status, which taps into the 5 domains of attention, immediate memory, delayed memory, visuospatial cognition, and language, and then the Dellis-Kaplan Executive Function System, or DCAFS, which taps into various aspects of executive functioning. And just to name a few of the tasks here, we have the trail making test and color word interference and verbal fluency tasks. Okay, so apologies for the slightly fuzzy table. This was just our, the ever important table one of the study, which just showed the simple clinical and cognitive characteristics of the sample. And the main reason I included this is just to show you that our participants from the late life depression group and the never depressed group were fairly well cognitively matched. Even though by design, the depressed group had higher self-reported and clinicians had symptoms of depression, cognitively they performed approximately the same or not statistically differently from those who were never depressed, which I think is important for the interpretation of the results. So what we did next, and this was the work of my former student, Livona Kalarko, who's now doing a PhD in medical biophysics. So we ended up going in and essentially calculating that contrast to noise ratio for the LC, as I explained a couple slides back. So this is essentially the same thing, but using one of our own images. Perhaps panel C is the most informative. So first, very briefly, we identified, again, the area where the LC is located, area of increased brightness right anterior to the fourth ventricle. We placed our over-inclusive mask here, and then we computed the ratio of the numeric value of the brightness in these voxels to the average brightness of this reference region in the pons, which again is known not to express any neuromelanin, so it's just sort of capturing the background signal. What we could then do is we could actually segment the locus coeruleus into 3 segments: rostral, middle, and caudal. And the reason we wanted to do that is, well, partially because the LC itself is quite a long structure along the rostral caudal dimension, but also because there were some postmortem studies indicated that perhaps the rostral subdivision of the LC might be particularly sensitive to aging early on. So you can see we're starting off with a fairly tiny structure and we're segmenting it even further, but we felt fairly confident in our segmentation given our approach. So what we then did was really started off with a simple case control comparison. So comparing the participants with late life depression to the never depressed controls on those metrics of contrast to noise ratio in those LC segments. And what we found was frankly a little bit underwhelming. So we actually found no differences between groups. There was one trend in the left rostral segment and then one near trend in the right rostral. So, in a sense, the rostral subdivision did show the most differences as expected, but none of this survived correction for multiple testing across the segments. And, we thought, well, maybe our sample size is too small, so maybe we're underpowered to detect effects. So we did some more extensive power analyses, but we essentially determined that we'll need in excess of 350 participants or something along those lines for these differences to become reliable. So overall, what we took away from this is that the classic contrast to noise ratio metric for the LC does not seem to reliably differentiate between those with late life depression and controls. So that's all very well, but we thought an interesting, still a relevant question was whether, despite this lack of differences, the LC integrity might still be associated with cognitive performance in this sample. So to test this, we actually collapsed across the late life depression and the non-depressed sample, mostly just, well, for a variety of reasons, both because the groups were well cognitively matched, but also to increase our power a little bit more. And rather than going, segment by segment and cognitive variable, we used the multivariate approach, a partial least squares regression to be precise, to try and map the relationship between the entire set of LC variables and the entire set of cognitive variables. And what we found was that there was indeed a significant association, one significant canonical variance, I believe is the proper terminology for partial least squares, capturing almost 73% of the shared variance between LC contrast to noise ratio and cognition across the R bands and the DCAPs. So this was very exciting to us, but we tried to kind of interpret it, and to do that we mapped this canonical variant back onto the original raw variables. So again, this is slightly fuzzy, so my apologies for this. I'll walk you through it. But essentially, we found that the relationship was a little bit more complex to interpret, but we saw strongest loadings from the left caudal and the right rostral locus coeruleus. And on the cognitive side, the strongest loadings were from the attention and memory domains, as well as a few from the executive functioning domain. So this study was really interesting, but it raised more questions than it answered, which is often the case in science. And we were, of course, welcoming of that. It just piqued our curiosity further. But the preliminary conclusions that we drew from this is that there is no reliable differences in locus coeruleus integrity, at least as measured by standard neuromelanin sensitive metrics. And there was a relationship between those metrics and cognition, but the relationship was a bit complex and difficult to interpret. And also it was not really specific to late life depression. So some important follow-up questions that we asked ourselves here, logically, hopefully, was, does this actually mean that the LC is completely spared in late life depression? Because that would be important knowledge as well, being able to rule that out as a region that might serve as a biomarker. So before we could fully rule that out, we wanted to sort of check if we might be able to find perhaps more sensitive indices of locus coeruleus integrity that could, in fact, differentiate from late life depression from never depressed controls, even in a small sample like ours. And then last but not least, the LC is a small, but it's a small structure, but the reason it's so important is partially because of those widespread projections that it has to pretty much the rest of the brain, a lot of other cognitively relevant regions. So we wanted to see, can we go beyond the LC and look at some of its connectivity to other regions and tap that for more reliable markers. So for this, we actually turned to diffusion tensor imaging as an index of microstructure. And we focus on the canonical measures of fractional anisotropy, or FA, or mean diffusivity, or MD. So again, very briefly, these are super familiar in the field and the literature, so nothing new here. But fractional anisotropy and MD are both typically computed for white matter tracts, but they can also be computed for smaller nuclei or gray matter structures, and they're particularly suited for structures that have kind of one predominant axis, such as the LC, which has that rostrocaudal kind of predominant dimension. So we were interested in extracting fractional anisotropy and mean diffusivity from the LC with the expectation that greater fractional anisotropy, similar to what you see in white matter, would indicate a healthier state of LC, whereas increased mean diffusivity would be the opposite. It would be associated with potential tissue damage, potential neuronal injury, and so on. But we wanted to go a step further and see if we can leverage tractography to reconstruct entire tracts connecting the LC to the rest of the brain. So again, what we can see on the right here is just the canonical white matter connectome reconstructed from diffusion imaging. So we wanted to kind of leverage a similar approach, but focusing specifically in LC. So far, so good. These are all very standard ways to use diffusion tensor imaging data, but there are some challenges, and they're all related to the small size of the LC coupled with the individual differences in anatomy between people. So the LC itself is only a few millimeters along some of its dimensions. So it really only spans a few voxels in some of the slices that we image it in. So, and those voxels are, of course, not always in the same place. So if we try to use traditional methods where we kind of co-register everyone's image to a common template, or we try to kind of find an overlapping mask of the region across people, we actually simply lose the structure altogether. So it became clear to us that we need to use a more individualized approach, working with each individual's native space. So trying to sort of honor each individual's anatomy to derive these individualized metrics of microstructure. And this is where the work of my former postdoc, Diana Valdez Cabrera, comes in. So she came into my lab with very extensive experience with diffusion-weighted imaging. So she took our neuromelanin sensitive scans. So this is just another view of that, and used them as essentially a localizer of the LC for each of our participants in their native space, again, honoring their individual anatomy. But then she took that and cross-registered it with our diffusion maps depicting mean diffusivity and FA, shown here on the left side. And those, the MD and the FA maps don't normally show us where the LC is. So by cross-referencing the two and keeping everything in each individual's native space, we were able to actually find and pinpoint where the LC was and pull out 3 different metrics. So again, the contrast to noise ratio, which is just coming from the neuromelanin scan. So that's consistent with what we did previously. We did this all in the same sample, by the way, the SENDEP study. And then we also, this is new for the study, we were able to pull out mean diffusivity and fractional anisotropy also for the locus coeruleus. In addition, Diana was able to, again, considering the individualized anatomy of each person, reconstruct the anatomical tract projecting from the LC to the hippocampus. And we picked that because it was a more feasible target rather than trying to build it out further to the prefrontal cortex. It is a more proximal target of projection, and it is actually known to be fairly cognitively relevant. So she reconstructed these tracts to the hippocampus via the thalamus, and we were able to extract, again, those microstructural indices, but this time for the entire tract. So we had mean diffusivity and fractional anisotropy for that tract, as well as fiber bundle capacity metric, which is a bit more of a tract-specific metric, capturing kind of the overall information carrying capacity, if you will, of this tract. And what we did next was very similar, again, to what we did in the first study. We started off with a simple case control comparison. And once again, no surprises here. We found that there was no difference between the late life depression group and the never depressed group. Again, this was kind of a repeat what we did before, so it was what we expected. I guess the only differences here, this is just the average CNR value for the entire LC, which then split by rostral, middle, and caudal. And then we looked at mean diffusivity and fractional anisotropy for the locus coeruleus, and we found that there were actually fairly significant, fairly large in size differences, where the late life depression group had both lower fractional anisotropy and even more so increased mean diffusivity. So both of these markers are indicative of potential loss of integrity or even neuronal loss, or at least neuronal injury to that small structure. And what about the projection tract? So interestingly, we did not see any significant differences there. There were maybe some very kind of nominal shifts in the same direction, but overall no significant differences. So the preliminary conclusion that we drew here is that there was in fact lower microstructural LC integrity in late life depression, while the projections from the LC to the hippocampus were relatively preserved. And lastly, this is not visualized here, but we also did look at the substantia nigra pars compacta here and found no effects when applying the same pipeline. The reason we did that is because we wanted to show the specificity of this to the LC rather than this, being more of a kind of generalized brainstem effect. So we did confirm the specificity to that little handful of voxels that we're fairly confident is the LC. And the next step, even though we didn't fully have the power to do that, we were urged by a reviewer to actually stratify by some of the clinical characteristics within our LLD group. We actually found that their results, those differences in FA and MD, were driven by those participants from the late life depression group who were not treated with antidepressants, whereas those who were treated, shown here the gray bars, had more of an intermediate position between the controls and the untreated group, but they were not statistically different from either. And then similarly, another kind of very apt question from a reviewer prompted us to try and tease out if there's a difference between those participants who experienced depression for the first time in late life versus those who may have had it earlier and were just experiencing a recurrence. And I should say that for the SENDEP study, the parent study, for the recruitment pipeline there, it didn't matter if this was like a first episode in late life or not. So when we looked at that, we found, again, kind of mirroring what we see with the antidepressants, we found that the effect was driven predominantly by those who actually had more of a persistent kind of recurrent course of the depression versus being in their first episode. So this is just kind of a summary of what I said. So, the way we interpret this is perhaps having these multiple episodes might do cumulative damage to the LC, or it is possible that reduced microstructural integrity in the LC is a potentially pre-existing risk factor that predisposes to more severe and chronic depression course. Okay, well, this is all very well, but what about cognition? So in the next phase, we quickly checked to see if there are any interactions between diagnosis and those LC metrics, and we found none. So we again collapsed our sample between those with late life depression and controls. This time we kept it univariate just for ease of interpretation, and we looked at each of the metrics individually and each of the cognitive dimensions individually, correcting for multiple testing. And these are the significant associations that emerged. So it's a lot of scatter plots, but just to summarize the overall pattern, we found that the only cognitive associations that we found were with FA, so nothing with MD. So we found that FA, especially in the projection tract, was associated with, so higher FA was associated with better performance, and this was regardless of diagnosis. It was consistent across those with late life depression and those without. And we did see also one association just with the word reading index for the LC itself. All the other associations were with the LC hippocampal tract. Okay. So just to summarize what we found in this study too, I think we're doing okay on time. We found that LC microstructure may in fact be altered in late life depression, particularly in those with persistent depression and those not treated with antidepressants. We also found that LC projections, at least to the hippocampus, appear to be relatively spared in late life depression, but they may instead be sensitive to cognitive performance more broadly in late life. So they might still be a useful biomarker for potentially finding those at risk for cognitive decline, regardless of the presence of depression. Again, our general conclusion is perhaps there's some promising evidence that the microstructure of the LC, rather than its sort of classic neuromelanin-derived metric alone, may actually serve as biomarkers of late life depression and cognitive performance in late life, respectively. So for the last little bit of my talk, I want to focus on the other neuromodulatory system of the brain, namely, well, there's more than that, but out of the two that I'm focusing on today, the other one, of course, being dopamine. And again, those two systems share a lot of anatomical features, as well as a lot of features in terms of the age sensitivity of their brainstem nuclei. And while what I showed you so far with late life depression was very specific to the LC, and in fact, we kind of went out of our way to show that it was specific to the LC, for this second portion of my talk, we are focusing on substance use disorders, where, of course, we set our sights on primarily on dopamine. And the reason for this is, fairly straightforward. There's a plethora of work that's associated substance use disorders, including alcohol use disorder, with differences in dopamine function. And again, some more recent emerging work has also linked substance use disorders with risk for subsequent cognitive decline and dementia risk. So given the abnormalities that are documented in dopamine function and that age sensitivity of the substantia nigra, again, thinking back to those postmortem studies showing both the LC and the substantia nigra having this buildup of neuropathology before other regions in the brain, we wanted to see if perhaps substantia nigra integrity could also serve as an index of age-related cognitive decline risk in adults with substance use disorders. So to test this, we used data from the Cognitive Dysfunction in the Addictions Program, or CDIA research program, which is a large collaborative effort that is just wrapping up now at CAMH. So just very briefly, it's a large interdisciplinary program with 7 interrelated projects, including a large clinical cohort, an embedded clinical trial, preclinical studies in rodent models, as well as neuroinformatics and healthcare utilization studies. They're all linked, all built around the central thesis that cognition, and specifically executive functioning, is central to outcomes in addictions and substance use. So I urge you to look this up. We just had our protocol paper published, and this was always intended as a resource to be made available, not just to CAMH researchers, but beyond that. So if you have any interest in substance use and cognition, please do look this up and get in touch. We are cleaning up the data, and we're very open to collaboration. For the purposes of today, I'll show a very preliminary analysis using data from this. So just very briefly, the inclusion criteria for this program is just basically we're recruiting adults, or recruited adults between the years of 18 and 60. We were able to recruit 400 at baseline. The only inclusion criteria was essentially they had to be seeking treatment for any substance use disorder. And again, this, for the purposes of this talk, this also includes alcohol use disorder. And we collected neuroimaging data from about half of them. And all quality control procedures considered, we ended up with about 100 with neuromelanin sensitive MRI. And everyone also completed a cognitive assessment. And for the purposes of today's analysis, I'm going to be using the neurocognitive index as sort of composite measure derived from the central nervous system vital signs battery, which is what we used as our main kind of cognitive screening, cognitive measure here. So this is ongoing work that's led by my master's students, Priya van Oosterhout, and this is just a preliminary look at the some of the clinical characteristics of our sample. Starting to rush myself, but I think we're doing well on time still. So this is just to kind of showcase some of the heterogeneity of the sample, which was very much by design. So you can see some of the main substances that were represented in the CDIA sample. Unsurprisingly, we saw the most participants meeting criteria for alcohol use disorder and cannabis use disorder, with substantial overlap between the two. And then that blue circle that says other, that's sort of like a grab bag of all kinds of other diagnoses, but it's primarily actually stimulant use disorder. So cocaine and amphetamine would fall under here, and there were a few participants with opioid use disorders as well. But overwhelmingly, we had an overrepresentation of alcohol, cannabis, and stimulants. And again, the substantial heterogeneity was very much by design in this program, and because the study was intended to partially capture a sample that's as naturalistic and as reflective of the clinical reality as possible. And this is really it's extremely common for people to be using more than one substance and to meet criteria for more than one substance use disorder. Okay, so we had data for about 107 participants, 30% female, of course, slightly underrepresented in terms of female participants, but still enough to do some potential stratified analysis later on. Average age was just under 40. And this is a very quick depiction of our neuromelanin pipeline as implemented for the substantia nigra. So very much the same principle as for the LC. What you see here is just a standard T1 weighted anatomical image, kind of averaged across our participants. Can't really see the substantia nigra very well. Maybe it does look a little bit darker, but you can see it so much better over here on the neuromelanin image. These bright spots here represent that. So again, we delineated those bright spots and we computed the ratio with a background region, in this case, the crus cerebri, which again appears dark because it doesn't contain any neuromelanin. And here we were able to actually compute those contrast to noise metrics on the voxel-wise level. Part of the reason for that is because substantia nigra is actually a bit bigger. It's still pretty small, but it is bigger than the locus coeruleus. So we were able to conduct some analysis that would allow us to essentially localize the effects of age and the effects of cognition within this search space of the entire substantia nigra. And this is a summary of what we found. And again, this is fairly preliminary, but we're excited about where it might lead us. So what we found was actually age effects that went in both directions, but in non-overlapping voxels. So the voxels that are shown in blue here were ones where higher contrast to noise ratio, presumably reflective of more neuromelanin, was associated with older age. And then conversely, the green ones was where older age was associated with lower contrast to noise ratio. So this was also independent of specific substance use diagnosis. When we controlled for that, it didn't substantially change the results. And it was also independent of more traditional metrics of global brain age, which are more heavily weighted towards larger structures, such as the cortex and larger subcortical structures. So this gave us some preliminary kind of hope that we might be capturing an early brain aging phenotype that's at least partially independent of these global macrostructural metrics of aging. And in the next step, we looked to see if cognitive performance itself, normalized for age, if it also maps onto contrast to noise ratios in the substantia nigra. And we found that it did, and specifically higher, so more brightness, higher contrast to noise ratio in the blue voxels there on the bottom was associated with worse cognitive performance. And it's kind of hard to tell by eyeballing it, but it does appear that there's an overlap spatially between the age effects, sort of like the blue voxels for age and the blue voxels for cognition. So that's what we wanted to investigate further. So we did like a conjunction analysis, putting these on the same overlay and looking for overlap. So we found that more than 50% of the voxels associated with cognition, remember that's already kind of normalized for age. So cognition, the voxels associated with cognition overlapped with those where greater CNR was associated with older age. So essentially, what that meant to us is that an older age-like neuromelanin or contrast to noise ratio profile of the substantia nigra was associated with worse cognition in this large sample of adults with substance use disorders. So again, this is exciting but very preliminary. So, what do we make of this? Like, what conclusions can we really draw? I mean, we could say for sure we have some preliminary evidence that substantia nigra structure is both age-dependent and cognitively relevant in substance use disorders. But is the evidence of accelerated aging of the substantia nigra in substance use disorders? I think this is fairly difficult to say, given that there are no published references for voxel-wise effects. So, you will recall that we saw there were divergent effects of age in distinct portions of the substantia nigra. Published studies have looked at the entire structure and have found kind of a similar inverted U-shaped curve to what we saw for the LC earlier on in the presentation. And this is sort of what we hypothesize is going on the level of the entire substantia nigra for substance use disorders. So we believe that the curve here has an earlier inflection point where in those who are a bit younger and certainly before middle age, there's elevated signal, but then later on they show kind of a faster loss of neurons. And we believe that these curves will differ for specific subdivisions of the substantia nigra. However, in order to test this explicitly, we would need a healthy control sample scanned on the same scanner, and some additional kind of clues as to whether this reflects accelerated aging could also come from trying to link some of these measures to molecular markers, perhaps known molecular markers of neurodegeneration or cellular senescence, validated in Alzheimer's disease and other contexts. So thankfully, we were fortunate enough to receive CIHR funding to do exactly that. So we're now recruiting a control sample, and this is a study I'm co-leading with Drs. Erica Vieira and Dr. Lena Quilty at CAMH. So we're hoping to recruit 150 adults without substance use disorders, so we could do like a side-by-side same scanner comparisons of those age-dependent trajectories of change, and ultimately map that onto executive functioning and other aspects of cognition, and molecular markers of cellular senescence and neurodegeneration, so that we can get a little bit more insight into mechanisms. And the title of this project is, we whimsically termed it CEDIA Age as a follow-up to the original study called CEDIA A. And we're looking specifically for novel markers of risk for age-related neurodegeneration, senescence, and cognitive decline in adults with substance use disorders. With the final goal, of course, of being able to identify those who might be particularly elevated risk for cognitive decline and dementia, and trying to direct interventions accordingly in line with the goals of precision medicine. So with that, I just want to acknowledge the trainees and collaborators who contributed to this work. I think there's many more that I can't put them all on one slide necessarily. I'll keep working on that. And of course, I'd like to acknowledge the funding sources. And with that, thank you also for your attention. Look forward to, I think we have time for questions, but if we don't get to any particular question, or even if you just want to stay in touch, please feel free to email me. And I should say this is an AI-generated summary of my talk, which I kept because I thought it was interesting and whimsical as well, although it might not be anatomically accurate. I thought it could be a nice visual to put on the screen while we're having our discussion. So thank you so much.
Thank you very much. Wow, for an entire presentation in one AI-generated pictogram, not bad. We'll open it up to questions, and please, if you're hybrid, don't hesitate to use the hand raise function or just speak up. While people think of their questions, what I'm wondering, Yuliya, is substance abuse and depression. There's a fair bit of overlap. And so how do you expect that to affect your neuroimaging findings, or maybe how do you plan on adjusting for that effect in your neuroimaging findings?
Yeah, that's an excellent question. So I mentioned that we were not looking to capture sort of pure groups of participants with just specific types of SUDs in our sample, and this was also true of other comorbidities. So we do have, as you suggested, we do have approximately half of our sample meeting criteria for depression or anxiety. So for sure that could add on. It could be an additional burden, although it does seem like when it comes to the substantia nigra, when it comes to depression, the results appear to be more specific to the LC, at least in later life. But what the way we're planning to deal with this is for our control sample, we're actually not excluding for a history of depression or a history of anxiety. So we're not recruiting, super controls, as they've been called sometimes. We're not recruiting people who've never had a touch of any kind of disorder, but we're trying to have a bit of a gradient of potentially some milder mood pathology, potentially some history of depression. So we're hoping that would help that be less of a confounding factor, if it makes sense. But other than that, I think we'll likely explore some studies just looking at more kind of cumulative symptom burden across substance use and mood, and see if what we're finding is specific to SUD versus more of a cumulative kind of symptom burden effect, if that makes sense.
Yeah, can I ask you to stop sharing your presentation so we can bring you back?